Hi there. So in our experiment, directly assisted by Joyce Regal and Kendrick Rubino and overseen by Lindsay Merrimanesh and Dr. Melissa Kaczynski-Collins, my lab partners and I attempted to evaluate and quantify the value of Drosophila as a model for Huntington's disease using the ELAV-Gal4 UAS-Q128 system. Huntington's disease is a neurodegenerative disorder which, upon development, causes a variety of psychiatric and cognitive symptoms. Not a lot has been discovered with respect to treatments and cures, but a polyglutamine repeat in the HTT gene is known to be the cause. This discovery mandates the use of model organisms in further exploration, which opens the door for a lot of debate as to which is the best. Drosophila have, however, broken through to the forefront of this type of research because there are strains that have been developed that use a neuron-specific driver, known as ELAV, to express a variety of polyQ repeats of different lengths. Also, because they are more physiologically complex than, say, yeast or C. elegans, it is a lot easier to see the phenotypic manifestation of the manipulated genotype. So basically, we used female flies with the ELAV driver and that caused expression of the Q128 repeat. You can see the genotypes listed under the materials and methods. And male flies that did not have the ELAV driver, which is neuron specific, and therefore did not express the Q128 repeat in the neurons like the female flies did. And we put 10 of each in a vial with a horizontal line about halfway up, which is demonstrated in the picture in the bottom center. And we would hit the vial down on the countertop and record the number of flies that got to the top of the vial after 10 seconds. And we did this 6 individual times. The number of Drosophila that made it to the top was recorded by using iPhone photos because it would have been really difficult to record the exact number after 10 seconds each time because they were constantly moving. So we would take a picture after exactly 10 seconds and then record the data that way. So the average number of each that made it to the top after 10 seconds after 6 trials is recorded in the chart in the bottom right hand corner. And so the average number of 3-week-old females that made it more than halfway up the vial after 10 seconds was 3.8 plus or minus 0.75, and the average number of 3-week-old males that made it more than halfway up the vial after 10 seconds was 6.5 plus or minus 1.76. And so we determined that Drosophila are in fact a good model organism for Huntington's disease, but that future studies should be, or at least with this system, should be done in females because they're the ones that are expressing the Q128 repeat, the ones that are showing a very visible lack of directionality, lack of mobility. And so we hope that further studies evaluate options for preventing development and expression of polyglutamine repeats in female Drosophila. And that's about it. Thank